All right, it is the top of the hour and we have close to 50 participants already. And I'm sure more folks will sign on shortly. First, I'd like to welcome Dr. Phil Halloran, who will be leading this this talk on this Sunday. And I'd like to welcome all the attendees from across the world for joining on a Sunday.
Thank you very much. It's an honor to be able to address this group, and I appreciate people giving Sunday. I hope I'll make it worthwhile. Can you see my screen?
Yes, we can.
You can hear me?
Yes, sir.
Incidentally, Kassem, shouldn't you be wearing a mask in an airport? Just
I am in a very, very isolated, secluded area here.
Okay, just checking, Kassem. I'm looking after you.
Thank you.
Molecular Microscope project is really part of a widespread series of studies involving HLA antibodies, the BAMF system. We're all studying the phenotypes of organ transplants, and it's all one. It's not competitive. And it's a pleasure to see my friend and longtime collaborator, Kim Soles, here. This project has commercial links, but it's actually largely been funded over the years by public money in the Genome Canada project. So we have links with One Lambda Thermo Fisher with a licensing agreement, and we're also doing research with Natera in the trifecta studies. And these are my disclosures. And this is the team. I'm very proud of this team. My main role is to go places and talk about what smart people do. So I'd also like to extend my gratitude to Luis Hidalgo in Wisconsin, who's working with us. Both my sons are involved in the project. They're associate professors here. So we'll talk about the MMDX system, new aspects of rejection, about diversity and T-cell mediated rejection, subthreshold antibody mediated rejection, DSA negative antibody mediated rejection, and the relationships to cell-free DNA. But I'd like to get you interested in exploring the parenchymal injury elements, which brings us right into all of nephrology to understand how the nephron behaves in response to damage. So the microscope diagnostic system. I love biopsies. I've always thought that the biopsy was where we would discover the truth. We discover mechanisms. It's also the site for definitive diagnosis. So avoid biopsies whenever possible, but optimize the utility when necessary. Biopsies if necessary, but not necessarily biopsies, to paraphrase some politician. And when I think about biopsies, and this is as a clinician and as a scientist, I think about the ways that biopsies can go wrong. There's sampling error where a sample doesn't represent the organ. There's intrinsic variability within the organ, like the kidney medulla. We can read the medulla molecularly, but we have to make adjustments for it. And then there's patchy things that go on, like atrophy scarring in some areas. When we're reading biopsies, we almost always use some degree of non-specific features, which we then submit to algorithms, clinical or BAMF rules or our machine learning algorithms to make them specific. And when we're assigning categories, you're putting patients in a bucket and not in another bucket. That is unreliable. So stuffing patients into one diagnosis versus another is really artificial because it creates boundary errors. And we all know about these boundary errors. And in addition, systems that rely on pattern recognition like X-rays and histology by a single individual have intrinsic inter-individual variability. So there's noise in those systems, which is probably to some extent not reducible, except by perhaps having 100 of them read it, which is not practical. And then in addition, our rules, our algorithms have errors. We started out BAMF 31 years ago. We had many errors and shortcomings, and we've always tried to correct those errors. So it's an ongoing learning response, partly because we're learning about the biology of the disease states. So the rules are always subject to reinterpretation and always should be. Then there are intrinsic limitations. The platform you're using, whether it's a microscope or molecular microscope, it will have limitations. You have to recognize those. You'll have weak phenotypes, and weak phenotypes like borderline or suspicious are important for the clinician to know, but they're kind of frustrating. What if the patient's been treated before you get the biopsy? What is the effect of treatment on the phenotype? What happens when you have multiple diseases? What happens if there are rare phenotypes? Now, you all have been taught things and you have experience with things, but you then see something that's outside your experience. The same thing happens with machine learning. If a disease state is outside of your reference set,